The pentose phosphate pathway. If you don't know what the pentose phosphate pathway is, I'll break it down for you. The pentose phosphate pathway is an alternative pathway to glycolysis, and its main goal is to produce NADPH and ribose 5-phosphate. In fact, it occurs in almost all cell types and tissues. This here is NADPH. Nicotinamide adenine dinucleotide phosphate is a cofactor in many anabolic reactions. It can play a role in protection against oxidative stress, however, it's primarily used in the reduction biosynthesis of fatty acids, cholesterol, neurotransmitters, and nucleotides. One of the products, ribose 5-phosphate, is an essential product of the pentose phosphate pathway, a vital part of DNA and RNA nucleotides. This all takes place in the cytosol, which is a fluid component of the cytoplasm. Let's take a deeper look into the cytoplasm to see the two separate phases that take place. There are two separate phases in the pentose phosphate pathway. The oxidative phase, followed by the non-oxidative phase. Let's take a closer look into the oxidative phase. As we know, oxidation is the breakdown of a molecule as it loses electrons. There are two irreversible stages to the oxidative phase. Stage 1. The glucokinase enzyme converts glucose that enters the cell into glucose 6-phosphate, diverting it away from glycolysis and shunting it into the pentose phosphate pathway. Once it has entered the pentose phosphate pathway, the glucose 6-phosphate dehydrogenase enzyme catalyzes the first reaction by reducing NADP+. This goes on to form 6-phosphoglucono-delta-lactone. Stage 2. In the second irreversible stage of the oxidative phase, the enzyme lactinase hydrolyzes the 6-phosphoglucono-delta-lactone to form 6-phosphogluconate, which is a 6-carbon sugar. When the enzyme 6-phosphogluconate dehydrogenase catalyzes the 6-phosphogluconate, it causes a carbon to be removed and carbon dioxide to be released. The electrons released from this reaction causes it to reduce the NADP+ back to NADPH, leaving us with a 5-carbon molecule, ribulose 5-phosphate. This ribulose 5-phosphate then moves into the non-oxidative phase. The purpose of the non-oxidative phase is to convert sugars to form ribose 5-phosphate, which is the sugar required to form DNA and RNA. The steps involved in this phase are reversible. Ribulose 5-phosphate can be converted into two molecules depending on the enzyme it uses. On the left, we can see that the enzyme ribulose 5-phosphate uromerase converts the ribose 5-phosphate. And on the right-hand side, the enzyme ribulose 5-phosphate 3 epimerase converts to xylose 5-phosphate. The first reaction uses the enzyme transketolase to convert ribose 5-phosphate to glyceraldehyde 3-phosphate, and the second reaction uses transketolase to convert the xylose 5-phosphate into cedoheptulose 7-phosphate. The glyceraldehyde 3-phosphate is further catalyzed by the enzyme transaldolase to form erythose 4-phosphate. Then, cedoheptulose 7-phosphate is also catalyzed by this enzyme to form fructose 6-phosphate. Erythose 4-phosphate is further catalyzed by the enzyme transketolase to form glyceraldehyde 3-phosphate, and xylulose 5-phosphate is catalyzed to form fructose 6-phosphate.